Okay, so I would like to begin first by thanking the organizers of the 2020 CCDS conference. And it would have been much nicer to see all of you in person, but I guess thanks to COVID-19, I think, although, I mean, the virtual conference could also be, could also serve the purpose. So I would like to thank Laura, Sangeeta and Andrea for kindly inviting me to talk at this special occasion. And today I'll be talking about some of the work which has been done in the lab of Professor Frysmuth. So me and as Laura mentioned, Sonia, we're working on this project for the last few years. And I will be talking about some of the things which we did. So to begin with some of the introductory slides. So creatine, so typically creatine can be taken by supplementary diet. So for instance, the consumption of meat, milk, or cranberries, for instance, could be served as an ideal source for creatine. And then creatine can also be synthesized inside the body. So if you can see on the left-hand panel, so typically in the kidney, so you have arginine and glycine, and the AGAT or the arginine glycine amide transferase catalyzes the synthesis of guanidinoacetate, which is an intermediate, and ornithine. And this guanidinoacetate inside the liver gets synthesized into creatine in the presence of the GAMT. Yeah. So once synthesized, this creatine can then act on multiple tissues and multiple target sites, such as brain or skeletal muscles, kidney, and heart. And once the creatine is has acted, the excess of creatine can then be reuptaken by the tissues. Yeah. So all these three important factors which are required. So the two factors, the AGAT and the GMT, which are involved in the synthesis, and the creatine transporter, which is involved in the uptake, are also present in the brain. Now, the creatine transporter inside the brain has been implicated in various functions. So people initially claim that it is involved in the energy metabolism. And then, of course, we know that it could also be involved in the process of neurotransmission, where it's a partial agonist for GABA A receptors. And the creatine is also involved in the process of osmoregulation, where in the hypoosmotic conditions, the creatine effluxes out, whereas in the hyperosmotic conditions, there is a shrinkage of the cells where the creatine influx is observed. So now, talking about the cerebral creatine deficiency syndrome. So on the left-hand side, you can see, so the two enzymes, the AGAT and the GMT, which are autosomal recessive, and the third, the transporter, which is X-linked. So these three, or one of these three, could be impaired, and as a result of which, the patient or the subject could suffer from various behavioral disorders. So there is a varying range of these symptoms. So one could suffer from seizures, intellectual disability, there is a delay in the speech, there could be digestive problems, and there could be movement disorders. So what could typically happen is inside the brain, when any of these three, that is the AGAT, GAMT, and the creatine transporter, if it's not functional, then we could suffer through reduced availability of the creatine or almost no availability of the creatine in the brain. And this could then lead to various clinical manifestations. So now the creatine transporter, if we focus on that, so creatine transporter is critical for reuptake of the creatine, and it is also critical for the uptake of this guanidinoacetate. And on the right-hand side here in the square, what you could see, so this is a diagram from Bryson and colleagues. And so these are the brain cells. And what you typically see here is a different cohort of brain cells. And typically GAMT, AGAT, and SLC6A8, which is a gene encoding the creatine transporter. So they could be present alone, or they could be present in combination in different type of cells. So what it provides us is there is a mosaic of expression when it comes to expression of these three, and the transporter is quite essential when you want to uptake the intermediate and the creatine itself. So the following slides, I will be focusing primarily on the transporter and how if the transporter is misfolded, how can that be rescued using pharmacological means? So the creatine transporter is a transporter which is belonging to the group of solute carrier 6 family of genes. And in our lab, we also study some other transporters such as the GABA transporter or the dopamine transporter and the serotonin transporter. And typically a point mutation or the missense mutations in these could lead to or is implicated in different types of diseases. And you can see that in the case of creatine transporter, there has been reports of various mutations, and it has been implicated in the X-linked mental retardation. And there has been no such clinical data, no such molecular data available. And this review comes from 2014. So we got quite interested. So what's exactly happening here? Because typically when you see a missense mutation, could it be a loss of function mutation? Could it be a gain of function mutation? So all these questions we start to ask. Now we will take one step back and we will ask how does this protein is formed? So typically the life cycle of all the secretory proteins, which will also include the creatine transporter, starts at the endoplasmic reticulum membrane. So this is the cytoplasm, and you have ribosomes in the cytoplasm, and this is a 661 translocation. So once it docks on the membrane of the endoplasmic reticulum, you will start to see the nascent protein getting formed. And once this protein is formed, it will undergo different quality control checks because you can imagine at the endoplasmic reticulum, there is a tremendous load of protein production and the quality control. So this protein will undergo the calnexin-calvindin cycle typically. So this is one of the quality control steps. And if it checks out, then the protein will go through the HSP relay of the quality control mechanisms. So if the protein is properly folded, what we call it the natively folded state, then the protein is ready to be exported in a COP2 dependent manner. So it will follow the secretory pathway of other proteins, and then it will be present on its final target, that is the plasma membrane. Now, what happens is if the protein is not formed properly, so then this quality control mechanism will stay in action, and this protein will then undergo a different pathway. So this protein will stay bound to this calnexin or HSP relay, and this will be then regarded for further lysosomal or proteasomal degradation. So if the protein is getting formed, it is allowed to have some access of helpers. If the helpers are not being functional, then it is cleared out from the system. So now this is a typical journey of a properly folded protein, which will follow this COP2 machinery and will eventually reach to the plasma membrane. Now, you can imagine that Leventhal proposed that if this process of protein formation is quite random, then it will take more time than the time of the universe to form the protein. Because typically a protein is around 500 amino acids long, and to synthesize such protein can take billions of years. So the process is quite regulated and it's quite ordered. And you can see here on this slide, two scenarios. We will begin from the right side. So this is the in vivo scenario where a nascently formed protein or unfolded protein wants to achieve a fully folded state, yeah? So the protein wants to achieve a state where the entropy is low, the energy is low, and the structural stability is quite high. And while doing so, it gets helped by other proteins or scaffolds, and that's what we call as the proteinaceous chaperones. And once this protein is getting help from these chaperones, it is allowed to form this fully folded state. Now, in vitro, situations could be very different because you don't have these folding proteins, which could be there in order to help the folding process that could happen. And so it could be that the unfolded protein can get stuck at different points in this energy landscape diagram. And as a result, you then have these mutated proteins which are stuck. So now one could use chemical chaperones such as DMSO and 4-PBA. So DMSO being an osmolyte. So what will happen is it will change the density of the environment for the protein, and basically it will allow the protein to fold properly. And in the case of 4-PBA, there are various mechanisms, but one of the mechanisms is the 4-PBA binds and stabilizes the protein structure at the hydrophobic sites. And while doing so, it allows additional time for the protein to fold properly. So if we revisit the a snapshot of the previous diagram, so if the protein is now harboring, let's say, a mutation, so it is still bound to the calnexin or this HSP relay, which are the quality control steps, then either after leaving these two steps, it can go out, or if it needs additional time, then different chaperones such as 4-PBA can still stay bound to the protein and allow it to give it further some time to fold properly. Now you can imagine at the ER, the protein concentrations are typically in the millimolar concentration ranges, which typically means that it's a very crowded place. It's the same as having thousands of people in a very small room, and everyone wants to dress properly, for instance. And so we come up with this idea, what if we relax the quality control step? So for example, we have inhibitor of this HSP70, such as pifithrin mu, or if we allow the protein to fold properly by introducing 4-PBA in the system, which could also inhibit the HSP70 and which could also bind to these proteins in order to help them to further stabilize the conformational states. So now this was the chaperone-COPII-exchange model, which we validated in number of publications. And before getting into the creatine business, I give you a snapshot of one of the human DART protein. And actually it was this project which motivated us to then consider the creatine transporters and the creatine transporter variants. So here on this slide, you see one such a case. So on the top panel, you see the wild type human DART. And typically for a transporter, if it's functional or not, so the conventional assay which has been established in the field is to perform the tritiated uptake. So this is a radioactivity labeled dopamine, and then we try to see if the cells take up the dopamine. And when we express the HEX cells, so these are human embryonic kidney cells, which are transfected with different variants of DART. And we ask a question, if we introduce these cells into the solution which has the tritiated dopamine, will the cells uptake the tritiated dopamine or not? So this tells us whether the protein is functional or not. Because if the protein is functional, then it has to reach to the plasma membrane. And after reaching so, it has to still do its job, which is uptake. Yeah. So what you see here is the wild type human dopamine transporter. And now it shows nice uptake. And now when we transfect the cells with these disease causing mutations, so these mutations are implicated in Parkinsonian infantile syndrome. So this is also known as the dopamine transporter deficiency syndrome, which typically affects the infantile and juvenile cohort of patients. And what typically you can see here is it's a loss of function scenario for all the mutations. And you see very marginal or almost no uptake for these mutations. So we talk about the specific case of B158F. So this protein shows almost no uptake, no measurable uptake. And if you see, so these are the HEX cells, and these are the confocal images of the HEX cells. So basically, trypan blue delineates the plasma membrane. And in the YFP, you see the protein. So when the transient cells are infected with the human DART, it nicely reaches to the surface. That's the end goal. And in the case of the mutant stays quite retained in the ER. So we were able to ask a question, can we rescue this misfolded protein by introduction of pifithrin mu, which as I said before, inhibits this one of the quality control steps. And we use a pharmacological chaperone such as noribogin, which selectively binds the protein and stabilizes it. And basically you can see that here, there was no measurable uptake, and after introduction of these drugs, both the drugs, we were able to see the measurable uptake. And we could validate this in vivo model as well, that the treatment resulted in rescue of the sleep pattern, which is typically impaired in the case of non-functional protein. So now this led us to ask, can we do the similar observations with the creatine transporter And here what you see here is a homology model of the creatine transporter. And this is based on the Drosophila dopamine transporter. And in the yellow or orange squares, spheres, you see all these mutations. So these are disease-associated mutations. And two of these are of particular interest because at this exact two positions, we also see its implication for Parkinsonian dystonia. So the dopamine transporter deficiency syndrome also covers these exact two spots with exact similar mutations. So then we asked whether the approach that we used before, can it be still used for the creatine transporter as well? And you can see here, so we asked a question, is it a loss of function mutation? So we tested 16 mutations and we saw that it's less or very low uptake that we observed with all these mutations, which could indicate that the mutations of these variants, these are loss of function variants. And then when we checked with confocal imaging, whether it is stuck at the ER or is it reaching at the plasma membrane, so most of these mutants were stuck at the ER. So this led us to the question, so these proteins, are they non-functional as a result of them being retained at the ER? Or is it that they are completely misfolded and the active site, which is here, the two sodium binding sites and the chloride site, is this site is also affected in the process of these mutations. So to ask this question, we treated the cells with 4-PBA. And here you can see there's a nice rescue that we observed in some cases. So the uptake was increased for these mutations, some of the mutations. And this was very encouraging for us that the proof of concept works in the case of the creatine transporter as well. And next we moved and we checked with the confocal images whether the mutant which was stuck at the ER in the cells, can it be rescued by treatment with 4-PBA? And you can see here in the case of, for instance, this mutation R391W, and in the case of P554L, we were able to see the rescue. So the protein does reach eventually to the plasma membrane, which was quite exciting for us. And we asked a question that this all sounds good. So here again, you can see that so these are quite technical terms. So the Km value is also improved. So the less the better. And the Vmax value, so the availability of the transporter on the surface, has also gone up. So basically the proof of concept works. And then we asked a question that this all sounds good for the cells, but can we rescue the protein in neurons? So neurons are the polarized cells, and you have dendrites and axons and different cell compartments where the protein has to reach and will the protein be functional in these such scenarios. And here you see one such case of the mutation P554L, where it's quite retained in the proximal dendrites and in the cell body. Whereas after treatment with 4-PBA, we see a nice rescue, yeah? So after Shaw analysis and measuring the area under curve, we could see a nice rescue, which is quite similar to that of the wild type. So all in all, what I want to say today is this folding disorders or the protein misfolding, which is observed in the case of SLC6 transporter and the creatine transporter, it can lead to various pathophysiological conditions. And the proof of concept that we have here with pharmacochaperoning approach works quite well. So we were able to confirm this in the case of dopamine and the creatine transporter in vitro. And we also confirmed this in the case of dopamine transporter in vivo. And this approach looks quite optimistic, as there could be drugs such as 4-PBA, which are already clinically approved. And these drugs, basically, since they already have passed through the rigorous process of clinical trials and are available in the market, so can they be repurposed? So this is a question that one could ask. And with that, I would like to thank all of you for listening. And I would also like to thank Sonia, who helped me immensely, my former supervisor, Freismuth, and my present supervisor, Thomas Hummel, and all the funding agencies. And with that, I'm happy to take any questions. Thank you.
Thanks so much, Dr. Kasture. I think this was great. I'm starting to add the moderators. So we have Dr. Laura Baroncelli, who's continuing as a moderator in this session.